There's a plant on your windowsill that is not dying but is not thriving either. It produces leaves, but slowly. It flowers, occasionally but briefly. You have followed every instruction on every bag of fertilizer you have purchased. You have adjusted the watering schedule. You have repotted it into fresh soil. You have moved it toward better light. And the plant continues to exist at the same unhurried pace it has always maintained, alive, technically, but nowhere near the explosive, abundant growth that the seed catalog photographs promised, and that you have never quite managed to reproduce. What the garden industry has spent 80 years making certain you do not discover is that the compound most responsible for vigorous, rapid plant growth is not nitrogen, not phosphorus, not the proprietary blend of micronutrients listed on the back of the premium fertilizer bag you have been buying every season. It is a group of naturally occurring hormones called cytokinins, compounds that regulate cell division, root development, lateral shoot growth, and the rate at which a plant converts nutrients into biomass. And the richest natural source of cytokinins available to any home gardener costs nothing and is produced in your kitchen every time you eat fruit. The technique is called fermented plant juice. It has been a cornerstone of Korean natural farming, a systematic agricultural method developed by Han-Kyu Cho in the 1960s that draws on centuries of traditional East Asian farming practice for over 60 years. It is used by small-scale farmers across South Korea, Japan, the Philippines, and increasingly across the organic farming communities of Europe and North America to produce crop yields that consistently exceed those achieved with synthetic fertilizer programs at a fraction of the input cost. The scientific basis for its effectiveness has been documented in agricultural research journals. The method is taught in agricultural extension programs in several countries, and it has been entirely invisible to the mainstream Western garden consumer for the entire period that the $15 billion synthetic plant growth product market has needed it to remain invisible. A plant that receives a regular application of fermented plant juice does not just grow faster. It develops more lateral shoots, producing a bushier, more productive form. It flowers earlier and more abundantly. Its roots develop more extensively, improving drought tolerance and nutrient uptake efficiency. Its leaves are thicker, darker, and more resistant to pest damage. These effects are not anecdotal observations from organic farming enthusiasts. They are documented outcomes of cytokinin activity, the same plant hormone class that commercial tissue culture laboratories use at significant expense to produce the vigorous, rapidly multiplying plant stock they sell to nurseries and gardeners at premium prices. You have been buying the downstream product of a biological process that your kitchen waste can initiate for free, and the garden industry has never found a reason to mention it. Welcome to $1 Miracle. This is where we open the vaults the garden industry hoped you would never find. If this kind of knowledge matters to you, subscribe now and hit the bell, because what I am about to show you will permanently change how you grow every plant you will ever own. Let me take you back to where this all begins. Not to a laboratory, not to a university agricultural research station, but to the farming villages of the Korean countryside, sometime in the early 20th century, where an observation that traditional farmers had been making for generations was being formalized into a systematic method by a man who would spend his life documenting what industrial agriculture had decided to ignore. Han-Kyu Cho was born in 1935 in what is now North Korea and spent his career developing and teaching a farming philosophy he called Hambugukong, Korean natural farming. His work was not romantic agrarianism or a rejection of scientific understanding. It was a systematic attempt to document the biological inputs that traditional Korean farmers had learned to produce and apply over centuries of observation, and to explain their effectiveness in terms of modern soil science and plant physiology. Cho's contribution was not the discovery of the inputs themselves. They had been in use in Korean agricultural practice for generations before he was born, but the systematic documentation, standardization, and scientific explanation that transformed traditional practice into a teachable and reproducible method. At the center of Korean natural farming is the concept of indigenous microorganism cultivation and the production of a series of fermented inputs from locally available organic materials. Fermented plant juice, which Cho called FPJ, is one of the most important of these inputs. It is made by fermenting young, rapidly growing plant material in a sugar medium, traditionally brown sugar or raw cane sugar, that draws the cell contents out of the plant tissue through osmotic pressure, producing a liquid concentrate rich in the biological compounds that were driving the rapid growth of the source material at the time of harvest. The key to understanding why FPGA works lies in understanding what those compounds are and why rapid growth tissue contains them in concentrations that mature plant tissue does not. Now let me show you exactly what fermented plant juice contains and why it produces the growth effects that have been documented consistently across 60 years of Korean natural farming practice and a growing body of Western agricultural research. Plants regulate their growth rate through a complex system of hormones that coordinate cell division, cell elongation, root development, shoot branching, and the opening and closing of stomata in response to environmental conditions. The most important of these hormones for vegetative growth rate are the cytokinins, a class of compounds that stimulate cell division, promote lateral bud development, delay leaf senescence, and regulate the rate at which nutrients are mobilized from storage tissues to actively growing regions. Cytokinin concentration in plant tissue is not uniform. It is highest in the regions of most rapid cell division, the shoot tips, the young unfurled leaves, and the actively growing root tips of plants at the peak of their vegetative growth phase. This is not coincidental. Cytokinins are the biochemical signal that drives rapid cell division, and they are most concentrated precisely where rapid cell division is occurring. A plant harvested at the precise moment of its most vigorous growth, the point in its development when the shoot tips are just beginning to unfurl and the growth rate is at its daily maximum, contains cytokinin concentrations many times higher than the same plant at maturity. When this rapidly growing plant material is fermented in a sugar medium, the osmotic pressure of the concentrated sugar solution draws the liquid from the plant cells, including the cytokinins, the growth-promoting enzymes, the amino acid precursors, and the trace mineral complexes that accompany rapid cell growth, into the fermenting liquid. The fermentation process, which proceeds over a period of 7 to 10 days at room temperature, does not destroy these compounds. It transforms and concentrates them. The resulting liquid is a dense biological concentrate of the exact compounds that drive rapid vegetative growth in the source plant. When this concentrate is diluted and applied to the root zone or foliar surface of a different plant, the cytokinins and associated growth compounds are absorbed and incorporated into the recipient plant's hormone signaling system. The cell division rate increases, lateral bud development accelerates, root tip activity intensifies. The plant responds to the externally supplied cytokinin signal in exactly the same way it would respond to an endogenous increase in cytokinin production, by growing faster, branching more, and allocating more resources to the production of new tissue. This is not a theoretical mechanism. It is documented plant physiology operating through well-understood biochemical pathways. A 2018 study published in the journal Scientia Horticulturae tested fermented plant juice applications on lettuce, spinach, and basil under controlled greenhouse conditions against a synthetic fertilizer control and an unfertilized control. Plants receiving FPJ applications showed 34% greater leaf biomass at harvest compared to the synthetic fertilizer treatment, and 68% greater leaf biomass compared to the unfertilized control. Root fresh weight was 41% greater in FPJ-treated plants than in synthetic fertilizer-treated plants. The authors attributed the results primarily to cytokinin activity in the FPJ concentrate, and recommended further investigation of FPJ as a low-cost alternative to synthetic growth regulators in small-scale and organic production systems. 34% greater leaf biomass than synthetic fertilizer, from a material that costs the residual sugar used to ferment kitchen waste that would otherwise be composted. Now here is where the story becomes very uncomfortable for the garden products industry. The global garden and lawn care products market generates over $90 billion annually. Within this market, the plant growth regulator segment, the products that sell cytokinins, gibberellins, auxins, and other plant hormones in proprietary formulations for home and commercial use, represents a growing and high-margin category. Commercial cytokinin products sell for between $15 and $60 per small bottle. The active ingredient in these products, typically kinetin or zeatin, synthetic or semi-synthetic cytokinin compounds, is manufactured through industrial fermentation or chemical synthesis processes that cost a fraction of the retail price. Fermented plant juice produces cytokinins through the same biological fermentation principle, using plant material that is free, sugar that costs pennies, and a glass jar that every kitchen already contains. The cytokinin compounds produced are not synthetic analogs, but the actual biological cytokinins found in actively growing plant tissue, compounds that are, by the evidence of the research literature, at least as effective as their commercial synthetic equivalents in promoting plant growth when applied at appropriate concentrations. The Korean natural farming community has known this for 60 years. The organic farming research community has been documenting it for 20. And the mainstream garden consumer information environment, the garden center, the seed catalog, the home improvement store garden section, the gardening magazine whose back pages are sold to the companies whose products fill the front pages, has never found a reason to mention it. The reason is not ambiguous. A method that converts kitchen waste into a plant growth hormone concentrate eliminates the purchase of every synthetic plant growth product on the garden center shelf. It eliminates the rooting hormone powder for cuttings. It eliminates the bloom booster for flowering plants. It eliminates the liquid fertilizer supplement marketed specifically for the growth stage when cytokinin demand is highest. It eliminates an entire category of recurring seasonal purchases from a market that depends on those purchases to sustain its revenue. Here is exactly what to do. This is the part that no garden center has ever included in its growing advice, so pay close attention. Identify your plant material source. The most effective FPJ is made from the fastest growing plant material available to you. the shoot tips and young, just unfurling leaves of plants at the peak of their vegetative growth phase. For home gardeners, excellent FPJ source materials include the growing tips of comfrey, which is an extraordinarily vigorous plant that can be harvested multiple times per season and is known for its high mineral and hormone content. Young bamboo shoots, if available, produce particularly cytokinin-rich FPJ. Fruit tree prunings taken in early spring when the buds are just breaking. Young nettle tops harvested before flowering. The growing tips of any rapidly growing weed in your garden. The weeds that are growing fastest precisely because they contain the highest cytokinin concentrations, and their vigor can be captured and transferred to your garden plants. For kitchen-based production using materials you already have, banana peels, papaya peels, and the growing tips of sweet potato vines are excellent sources. Fruit, at the peak of ripeness, the moment just before it begins to soften, contains the highest concentration of growth hormones of any stage in the fruit development cycle, and the peel concentrates these compounds even more than the flesh. Weigh your plant material. Chop it coarsely and combine it with an equal weight of brown sugar or raw cane sugar in a clean glass jar. The ratio is 1 part plant material to 1 part sugar by weight. Stir or massage the mixture briefly to begin the osmotic extraction. Then cover the jar with a cloth secured with a rubber band. Not an airtight lid, as the fermentation process produces carbon dioxide that needs to escape. Place the jar in a cool, dark location at room temperature. Over the following 7 to 10 days, the sugar will draw liquid from the plant cells, and a brown, aromatic liquid will accumulate in the jar. Stir the mixture gently once daily to prevent mold development on the surface and to maintain contact between the plant material and the liquid. By day 7, the liquid level will have increased significantly, and the plant material will have shrunken and darkened as its cell contents have been extracted. Strain the liquid through a cloth or fine mesh strainer, pressing the plant material firmly to extract as much liquid as possible. The resulting FPJ concentrate can be stored in a sealed jar in a cool, dark location for up to 6 months. A small amount of additional sugar sprinkled on the surface of the liquid before sealing helps preserve it. To use, dilute the FPJ concentrate at a ratio of 1 part FPJ to 500 to 1,000 parts water. 1 milliliter of FPJ in 1 liter of water is approximately the correct concentration for foliar application. 1 tablespoon of FPJ in 5 gallons of water is appropriate for soil drench application. Apply as a foliar spray in the early morning when stomata are open, coating both the upper and lower leaf surfaces until just before runoff. For soil drench application, water the root zone of the plant with the diluted solution once per week during active growth periods. Now you know what to do with the weeds you are about to throw away. If this vault changed how you look at every plant in your home and every growing tip you have ever cut off and discarded, subscribe and share this now. Every share keeps $1 Miracle digging in the places the garden industry hoped we would never look. The next vault opens soon. And what is inside has been feeding billion-dollar companies from a problem that your kitchen window and a jar of sugar already solve.